Actually, why don't I do this and then I can just share.
Oh yeah, we had to do that 3D picture thing and answer the questions on it.
Yeah, it had the side-by-side comparison. What'd you think of
Yeah.
that? Were you able to use the Flash version or did you have to use the just the text version?
I used the Flash one. That was
Oh,
actually, I didn't mind that because all the answers are right in there. It was,
yeah, they are. They're right there. And the Edulastic was just a way for me to record the answers. So you take your time, put them in, and some of them are open-ended, like they're short answers. So I go through and I check all those, but it is pretty straightforward. Are you using a Chromebook with that?
yeah,
And so did my directions work or did it automatically work on your computer? You had already set it for Flash.
I just clicked the link and it opened up.
Oh, cool. Okay. Because in the upper, you can see my screen, right?
Yeah.
This little lock icon, that has this thing on there that for some Chromebooks, they are blocked. And so the students can't use the Flash and they don't know to change that setting, or they may not even have privileges. I'm not even sure. So I had to make sure I had both versions. But the fact that you use the Flash version, that's definitely a benefit because, oh, see, what did I do?
Yeah,
Select.
mine didn't do that, so I guess it was already unlocked.
There you go. Yeah, well, it said it would say ask by default, and that's what it would do. It would have locked it like that. But seeing this is what you want to see because you can tell, like for example, when you get into this here, like that screams the huge difference between mitosis lining up down the middle and then meiosis lining up on both sides. Let me just see if there's anyone else. I got Mallory. Okay. Go back to that. I was here, and then I was here. So if you see anything on like any type of like unit check-in, you want to make sure that you really grasp that the difference behind what happens in mitosis and meiosis. And meiosis happens twice. It's like a two-step process. And so the first step process, this is the difference. The fact that the chromosomes, and so let's just make sure we understand what we're looking at here. The color, all right, and the length are what you want to look at. So the fact that we have orange, actually, let's go back. That's the better picture here. So the fact that we have a long orange and a small orange and a long blue and a small blue means that in the original cell, do they even show a chromosome? They don't. I would have shown one more step between this, but in the original cell, there would have been one long orange and one long blue. And those would have been homologous chromosomes. They would have had the genes, or yeah, they would have had on those chromosomes genes that code for particular traits. And there would have been one small blue and one small orange. And those would have been homologous chromosomes. Same thing. One chromosome from mom, one chromosome from dad, both code for the same sets of genes. Where you see the two colors in the X-looking shape is the sister chromatid. So this is the one chromosome and its copy. Does that make sense?
Yeah.
Okay, so when you get to mitosis and the difference between meiosis, you could see that in mitosis, when this cell splits, the two new cells will indeed have one long orange, because one will go here and one will go there, one long blue, one's right over here, and one long blue here, one small blue here, one small blue, and one orange, small orange. And so each one will have the exact same information. But in meiosis, because they don't line up in a line and they line up on opposite sides of this division, you end up with two cells that are not functional. Even though they have the same number of chromosomes, 1, 2, 3, 4, this one has the chromosome for, let's say, dad, and it has the exact duplicate of dad. In this chromosome, you see what I mean? It doesn't have the other chromosome for mom. It has dad's chromosome and its duplicate. It doesn't have the other homologous chromosome.
Yeah.
Makes
Yeah.
okay, because then what happens is these two will then divide just like this. So when mitosis, I don't have this in like a PDF, but so when meiosis occurs the second go around, all right, what you'll end up having is at this same stage, instead of having this cell at this stage, you will have each of these cells will be at the metaphase stage. And what will happen is this cell here, which will have 2 long blues and 2 small oranges, will divide just like it does in mitosis, so that you'll end up with 4 cells. You're going to need to see this. So let me put this on here. Let's go here. I'm doing all my law stuff here. So we will go to a new document and over here. Okay. So if you have, so let's do this. We're going to do this one. And that works enough. Actually, let me go back because that's a little better. All right. So we're going to put, well, they had orange, so we'll use orange. So we've got in my meiosis, we're going to have one long and its copy, and we have one small and its copy, and then there was a blue one, right? Is that they used? Yeah, I think so. And then we've got, we'll put the other small one up here, and then we've got this. All right, that's in mitosis. So this represents metaphase, and that's in mitosis. All right, and then metaphase, because in meiosis it happens twice, they call it metaphase 1 and metaphase 2. Sorry for my chicken scratch. In this one, we are going to have, on this side, we're going to have this, like this, and then it doesn't really matter. I guess it can go on. There's no rhyme or reason to which side they line up. So we can have this one here, and then we have the orange, and we've got this one lining up here, and this one lining up down here. So now what happens is when these cells divide, so when this cell divides right down the line through here, you're going to have one going this way and one going that way. And you know what they're going to end up as, right? Each one's going to have one of these.
Yeah, they have to be the same level,
Yep.
right?
So in metaphase and meiosis, because they're lined up on both sides instead of down the line, and this cell splits like this, and this one goes this way, and this one goes this way, you're going to have 2 long orange instead of 1 long orange and 1 long blue. You're going to have 2 long orange on one side and 2 blue. That doesn't even resemble what the original cells would look like. But what then happens, so now this cell basically splits to here. So let's go ahead and draw those in. Let's do this. So we basically end up with this, and we end up with this. So what would that look like? That would look, in this picture, we've got this. We've got this. So then in metaphase, now, so there's going to be a whole bunch of process, but metaphase is going to happen again. It's going to go through all this stuff, and metaphase happens a second time. And when it happens the second time, we'll draw orange. In the second time, it lines up the same way it did in mitosis. So now, in the second go around, they do split up right down the middle. Okay? And you end up with 2 cells, actually 2 cells each. Well, let's draw. So now you're going to end up with one this way, one here, and one here. Are you following so far?
Yeah.
Okay, so then we end up with, all right, and this will be quick now because we only need, that blue works for me. So we'll end up with,
Would there just be one in each thing then?
yeah, so that's the important thing. Good, I'm glad you said that. So let's make sure I got this right. So there's going to be one long, one short, and one long orange, one long orange, one short, one short. These are the sex cells. And so this same process, now here's what's so cool about this. This same process that we just did, okay, so let's say this is the male, right? So these are the male sex cells. Now for the female, you got the eggs. Let's just say it was the same cell and this was the female. There's no rhyme or reason that the orange has to be on the left-hand side. I just did that. So the orange ones could both be on the left-hand side and the blues could both be on the right? I mean, that's a possibility, right? I mean, there's no rhyme or reason. Remember, you've got little centrioles, right? And they've got those spindle fibers and they're moving all this stuff around. They don't know how to organize which one's on which side. They just know that I have to have one on one side and one, homologous and its sister chromatid on the other side. So this is where genetic variation comes into play. Imagine in the mom that these chromosomes were flipped in this particular arrangement. That would result, all right, in, instead of having one orange and one blue like we have in here, we would have both two blues in there, right? Are you following or no?
Yeah.
Okay. So now if the moms are, let's say we kept it with blue. Let's say so we're saying we're going to switch this. We're going to take this. Go back. We're saying we're going to take this and just imagine we flipped it to this side and we took this one and we flipped it over here. That would make this side here a long blue and a small blue. And then both of these on this side would be a long orange and a small orange. So now if this is the mom's egg and that's the sperm and they get together, then you're going to end up with a child that's going to have one blue and one orange. In other words, that child would have gotten, let's say blue represents dad and orange represents mom. And this is the current mom and the current dad. And what they're showing here is in the sex cell for the dad, the longer chromosome came from this dad's father, and the blue chromosome came from that dad's mother. But in the mom, those sex cells both came from the mother, so to speak. But another arrangement, so every time the eggs drop, they're all dropping different, and this arrangement can be different every single time. The same with the sperm as well. Now imagine there are 46 pairs of these. So imagine 46 of these chromosomes going down in this image right here, and then arranging number one, the pair number one, flip pair number 4, flip pair number 8, flip pair number 23. Imagine how many arrangements of those 23s you would be able to flip blue and orange. Do you see what I mean?
Yes. Yeah.
Okay, and so what that does is that creates all of the genetic variation. You remember when you did Punnett squares?
Kind of.
So you did these boxes like this?
Yeah.
Okay. Well, don't panic because you're going to understand this now. You know why? Because you see what I just did? How I said the possibility of a long blue and a short orange. This egg also has the possibility of producing a long orange and a small blue. So let's just say for simplicity here, I'm going to move ahead, all right? Let's just say that on this chromosome, at this location, I hate when that happens. Oh, it does that all the time to me. It's because I write too small. Yeah. All right. So let's say at that location, there's a gene for eye color, and the orange represents the dad. So this is a person, and they have chromosomes from their mom and their dad. And the orange represents the chromosome that this person received from their parent. And on this spot on this chromosome, there happens to be eye color, and that eye color is a capital B for brown eyes. Okay? Now, is that making sense so far?
Yeah.
Okay, now, that dad, unbeknownst to him, on the chromosome he had from mom, which is the blue long chromosome, all right, on that chromosome, again, at the same, mom actually gave him a little b. So even though this dad has brown eyes, so we can say is brown, we can say little b is blue. This dad is a carrier for blue eyes. Making sense so far?
Yes.
Okay. Now, mom, okay, she's got the same. I don't know why I keep doing that. So she's got same traits right there. And she also, mom, so this person here, all right, what we'll do here is we're just going to erase these words mom because that'll be confusing. All right, it's just going to have to deal with it. PDF annotator, unnamed PDF. All right, let's see what happens. Let me move something. Don't do that to me. All right, let me switch over and just see if switching out of this stops that. I think we're okay. We'll see that. Okay, well, I'm going to have to explain to you then. So let's just say, because I'd have to redraw. That's what it looks like it's going to make me do. So let's say over here on this mom, she has, let's say it's a capital B here. So she also has brown eyes. And on the chromosome she inherited from her dad, it's a little b. So in your Punnett squares, what you were doing in Punnett squares is you were saying, it's possible for dad to give this a sperm cell, which has the capital B, but it's also possible for the dad, all right, we'll assume this represents the dad and this represents the mom. It's also possible because the dad has a carrier of blue eyes on his chromosome he inherited from mom, that he could inherit or he could give to the child this particular sperm cell, in which case it has a little b. That's what went in those boxes. You put either big B or little b in your top box. I'm going to try to, let me see if I can just do one more. So in your box, you would have had this, and you would have put, for dad, he could have maybe given this one or this one, because it could have been either or. Mom, so I can't, I'm sure this is not going to do anything. It's going to do the same thing. Oh, capital B, lowercase. Okay, mom can do the same thing. This could be the egg that mom deposits that goes with that sperm, or the egg that drops in that particular woman's cycle could be this one, which can go with this. Or this one here could go with this sperm, or this egg here, if that's the case, could go with that one. And so when you did your Punnett squares, that's what you were doing, is you were going ahead and you were saying, what are the chances that these sperm and egg, if they get together, are B, capital B, capital B? What are the chances that it'll be capital B, lowercase b? In other words, what are the chances that this one here goes with this one here? You see what I mean? What are the chances that this one goes with this one? That's what's happening in this box. And then what are the chances that dad donates one of these others? Because there's four. Remember, there's 1, 2, 3, 4. It's not just two. Each one splits. So dad's got, out of one cell, dad's got 4 gametes or 4 sex cells. So he's also got another capital B, because remember, we didn't do this side here. If we had done this, this would be a little b. And I used this one over here, but because now I made a mess out of this. But if we really did this, this should be capital B. If we erase, let's get this out of here for a second. So you can see capital B, small, so they're the same. They're the same here. So dad has an opportunity to give 4 of those, just like the mom has an opportunity to give 4. That's why there are 1, 2, 3, 4 boxes, because you don't know which of the four, you don't know which of these four dad's going to donate, and you don't know which of the four mom's going to donate to the offspring. And that's why the Punnett square has 4 boxes, because you know this is the possible things mom and dad can donate. And then in the boxes, you try to figure out what's the probability that your child is going to be capital V, capital B. What's the probability that, and that's brown eyes, by the way. What's the probability that there'll be lowercase b, which we said is blue eyes? And what's the probability that there'll be capital B, lowercase b, in which case they'll have brown eyes, but they'll be a carrier of the blue eye? Are you seeing a little bit of the connection as to why meiosis, where it ends with 4 cells, is the reason why Punnett squares have the 4 boxes?
Yeah, I put the square for like last year, and I didn't really understand what they were for.
Right. You just probably thought it was a magic box of things that predicted what the baby could be.
All the letters.
Yeah. So now what I want you to do is I just want you to start thinking, okay, at the end of meiosis, all the cells, the 4 cells that are going to be generated at the end of this process, number one, none of them have enough chromosomes to function. They all have half of what the original parent cell had. Think about it. Parent cell, because this is already duplicated. So this parent cell, before we started, it's hard doing this because you have so many little circles and stuff, but this parent cell before we started had 1 blue. Let's go. Had 1 blue, had 1 small blue. I'm not going to worry about the small chromosomes yet because there's just too many. And 1 orange and 1 small orange. So we're not going to worry about, let's just forget for a second that these chromosomes are even part of this. Let's just focus on the blues. Actually, no, you know what? Focus on them all. It doesn't really matter because the cell shows that. So here you go. Here's the original cell. This is what it looks like in meiosis, in the metaphase stage of meiosis. And this is the end result. And so these are sex cells because they contain half, 1, 2, as compared to 4 chromosomes. They don't have the chromosome and its homologous partner. They just have the 1 chromosome and that's it. That's why you need the egg and the sperm to get together to make the whole. And the idea behind the Punnett square is to give you this idea of the variation, like what is the probability that this sperm cell hooks up with an egg cell that's just like it. Make sense? A little?
Yes.
Okay, because that's really important because that's where you're headed. So what I'm trying to do is I'm trying to front load some of the Punnett square stuff that you're going to see on Friday. And that's all we do. We just do this basic Punnett square stuff. But it's really, really important that you understand that the end result of meiosis is to generate the sex cells, which have half the genetic information, as opposed to mitosis, which purpose is to not do that, but to generate a duplicate cell because you're trying to maintain homeostasis by repairing the body and so forth. So you need functioning cells in mitosis. So they have to have all the genetic information to be able to do that, not half of the genetic information. And the way meiosis gets away with it is by being very crafty with how metaphase occurs in meiosis. It occurs side by side in the first step of meiosis, and then it goes to the similar setup the way it is in mitosis. The only problem is by that time, what ends up resulting are 2 cells that have half the genetic information, which we call gametes, as opposed to the other cells in your body, the way they divide, which is mitosis. It's a lot. Biology is it's fascinating though. This part's cool because you can kind of get an idea now of, if there were 46 pairs of these things or 23 pairs of them, that's a lot of chromosomes, a lot of variation. And I don't know if you noticed, but this was a question yesterday, and I don't know if can I zoom in. If you look really carefully, you'll see that part of this blue chromosome is attached to the orange, and part of the orange is attached to the blue. Do you see that?
Yeah, so that's like what you were saying, right?
Yeah, so what happens now, only to further confuse you, because that's what we love to do, is, and I'm not going to draw this because that'll confuse you, but what happens is sometimes when the chromosomes get in this side-by-side piece here, what'll happen sometimes is this piece here will exchange with this. So now you have genetic variations. Remember, let's say on mom's chromosome, remember, see, we said mom, this blue one is mom, and she's a lowercase b. Suppose mom had another trait for hair color. Oh, come on. You're really making that there. Okay, so let's say mom had another trait and it was hair color and it was located down there. And she had a capital B for brown. Okay. And let's see, so that'll be capital B there. And the chromosome, the other chromosome that mom has for that particular chromosome, let's say she was a carrier for blonde hair. Oop. Yep, that's right. It's going to do that to me again. So she's a carrier for blonde hair. So what this means now is that on that same chromosome, there's more than one trait. There happens to be the trait for eye color, which happens at the top half of the chromosome. And down here, there's another trait that happens to fall. Let's just make believe this is chromosome number 3, right? And on that same chromosome is the trait for hair color. And this particular individual has a capital B for a totally different trait, and that will say represents brown hair, and the lowercase b represents blonde hair. And so this particular individual has brown hair because they have the dominant trait, but they're carriers of the blonde gene. Make sense so far?
Yes.
Yes. Okay, so now under this pretense, we would argue that if my child has blue eyes, then they also received the brown hair trait, right?
Yeah.
Okay, all right. So, okay, that's if they, so if the child, let's just say we don't know what dad is, all right? Let's just make an assumption. If the child receives this, we'll say this is the mom. So this is the egg. So if the child receives, or the to-be child receives that particular egg, then, and we don't know what dad's makeup is for these two traits, we could safely say, considering this mom, that child will have the chances of having brown eyes, brown hair, and blue eyes, possibly, depending. Okay, now, if they receive this one, then the child has the potential of having brown eyes, but possibly having blonde hair. Okay, what's really cool is if what I showed you right here, if this crossing over occurs, which is what this is called, what that means is that this segment would have swapped with this segment. You see? And then what that meant, what that would mean is this would not be a little b, it would have been the capital B. And this would not be a little capital B, this would have been the little b. And so you would have ended up with a blue eyes and blonde hair, and brown eyes and brown hair, not because that's how the chromosomes were, but because there was genetic material that was literally pinched off one chromosome and placed on another. And what that does is that adds another element of genetic variation. In other words, we could go through and we could make a prediction based on what we know your genetic makeup is, and we could say this is what are all the possibilities. But once you throw in this idea that chromosomes, when they're too close to each other, can all of a sudden swap parts of chromosomes, you add an unpredictable component of variation that a geneticist couldn't account for. And does that make sense?
Yeah. A little.
Well, okay, so I'm probably going a little outreaching a little bit too much, but this is where we're heading with the genetics. So
Yeah,
you can watch it over and over. They're all recorded, so this is a good thing that we can just put that aside on